He's director of the Brain Dynamics Lab in psychiatry at Stanford. He's a professor of computational neuropsychiatry, and I've known Dr. Sagger for a long time, too long probably. He's the person I always put on a grant as a machine learning consultant because he's really an expert at the forefront of the field and using machine learning approaches for neuroimaging data analysis. And he's been for over the years inventing and disseminating a lot of tools to look at brain dynamics in and fMRI data in particular, really kind of exciting applications, bringing in the theory and methods from engineering and computer science, and just really at the forefront of this. So if you're thinking about doing anything with brain dynamics and neuroimaging data, this is really like what they call like leading edge, cutting edge kind of stuff. So it's really interesting, and I'm going to give him all the time to work on it. But just to give you some background, Manish has received many R1s and Directors Pioneer Awards from NIH recently. So obviously they think this work is exciting and important. So we'll be here, and I think this is going to be a very interactive kind of hands-on thing. So the first part is really describing theory and talking about methods in general. After lunch, when you get to know him much better, we'll do the hands-on workshop where you can really ask even more detailed questions. So don't be shy and feel free to jump in. So please join me in welcoming our speaker, and thanks again for.
Thank you. Thank you. Hope you can hear me okay. Yeah, good. Fine. That was a beautiful introduction. Obviously, I don't deserve any of it, but thank you. Okay, so today I'll talk about how we use TDA, Topological Data Analysis.
Can everyone hear in the back?
Okay.
Yeah, okay.
Anybody here has done TDA before? So I can say anything I want. Great. Okay, so I'll give you a brief background, and then we'll dive into how we're using TDA, and at the end I'll save some time for some of the new methods we developed. You can ask questions at any time. Okay, so coming from psychiatry department, I have to give this spiel, I guess. So we need good methods to study brain dynamics because, as you may already know, it's very hard to anchor mental health disorders into something that we can biologically measure and study. So just to give a comparison between something like diabetes versus any favorite mental health disorder of yours, etiology and pathophysiology, meaning what causes the disease and how it affects everything else, is complex in both of the cases. In case of diabetes, we have this beautiful, I don't know if it's beautiful, but it's a very good signal, hemoglobin A1C, that we can measure, or some other signals like that. So we know exactly what is happening or at what stage you are in the disease. Mental health, not so much. We have just a cluster of symptoms. And this hemoglobin gives you this idea of a nice biological feature that we can then use to do precise diagnosis, management of the disease, and so on and so forth, and develop good treatments. But in case of non-biological symptoms, like in case of mental health disorder, we have a mess, kinds of fun stuff. So how do we solve this? So one idea out of 4 things that my lab is working on, one of the ideas is, can we use brain? Because actually mental health disorder is apparently brain related. So can we use the brain? And somehow, can we pick up the transitions of brain activity? So not averaging data in space, time, or across people, but can we let the data tell its stories, kind of an old cliche in machine learning, but can we use that in a way that we can capture the shape of the brain dynamics? And that's what I'll talk about today, mostly. But the other 3 techniques are this, which I won't talk about, but to anchor psychiatric disorders, we believe that the main challenges are obviously measurement noise. And we were just talking about global signal being some people's favorite, some people hate it, but God knows what it is. So there's a lot of measurement noise in the fMRI data. I don't know if they're recording me, but sometimes F stands for fake MRI. So we don't know, right, what the heck we're really doing. And there's obviously limited use of computational modeling, and I'm very excited about that part too. So if you're interested in biophysical network modeling, love to chat more about that. More and more of our experiments we're trying to do outside of the lab, so using portable neuroimaging, happy to chat about that too. And then with Lucina, we have this R1 where we're trying to validate the nosology itself, the art of itself, using, again, data-driven methods. I'm very happy to chat about that too. But today I'll mostly focus on the first part because of the time. So what do we do as neuroimagers when we have so much noise? We love averaging. So we average. And obviously it's not true anymore. I don't have a pointer, I guess. But yeah, so it's not really
true anymore that you're averaging over everything, but most of the studies used to be at least in this average space. Nothing wrong with it. The only problem is that we end up losing um, some idiosyncrasies of people. And I'll talk about that in a little bit detail. But more often than not, we are now looking at or moving towards this direction. So rather than looking at group data only, we're looking at individual participant data or within session data. So we're trying to capture those idiosyncrasies. We are also going away, thankfully, from whole brain. Again, nothing wrong with it. Maybe there's some good signal there too, but we are letting the data tell its story by going all the way to single voxel analysis. For example, you may know the famous work in Midnight Scan Club from Nico's lab, where they have this individually defined brain networks based on 10 hours or 5 hours of resting state data. But what about time? And also there's a lot of work that goes from looking at sliding windows or even single TR resolution stuff. And that is very critical, I think, is because time might hold the key to looking at psychiatric disorders. So if you have very fast dynamics, you could potentially pick up symptoms of ADHD versus depression or rumination for slow dynamics. But the best idea would be to just go, so hopefully you'll agree with me, and I'd spend too much time on this animation, but the best idea is to kind of stay here, right? So let the data tell its story. So if you guys are motivated enough, let's just do it. And we are calling it precision spatio-temporal mapping just for fun, but it's basically going close to the acquisition data as possible. So how do you do that? One method, there are many methods. Under the umbrella term of TDA, topological data analysis, there are many methods. One of them is called mapper. Basically, you're mapping data in some way. But there's another methodology, precision homology. I'll talk about that in a little bit more detail in the afternoon if you're interested in that. But this talk is mostly about mapper. So the idea with the mapper is if you have any kind of data, so in this case, it's a very simple example of a 2D circle. The goal of mapper is to find out what's the shape of the data. So in this case, yeah, it's a whole, big deal. But it's actually a big deal when the data is very high dimensional. And you want to know the shape. And the idea is once we know the shape, then the noise doesn't matter as much, hopefully. And we can kind of predict things that we have not seen before. So this work was originally credited to Gurjeet Singh, Facundo Mamoli, and Gunnar Carlson at Stanford. Carlson also was my mentor when I was a postdoc at Stanford. So all credit goes to them for this amazing work. And we're kind of extending it to neuroimaging data. It was one of the first papers by my grad student, Caleb Denise, where we showed how mapper can be used for neuroimaging data. So let me just give you another example of how mapper works. So imagine a three-dimensional data set like a trefoil knot. So the idea with this data set is if you're an ant for some reason and you start walking on it, you will never fall off, meaning the shape is a closed loop. Now it's a three-dimensional data. We took it for a reason as a three-dimensional data. And the idea is how can I take this, put it in a two-dimensional data, so do some dimensionality reduction, but maintain or capture the shape that it's a closed loop. And that's one of the challenges of mapper. So just like any typical dimensionality reduction technique you do, you just go from, let's say in this case, we went from 3 to 2 dimensions, and we basically got rid of the z dimensions, so now we only x and y. And the minute you do that, you lost this idea that it's a closed loop. Now it's like this point cloud of crisscrossing. And that's where your typical dimensionality reduction stops. Right? PCA, T-SNE, whatever you like, it stops there. So the idea is how can you capture the shape? So we go 3 more steps to capture the shape, and the shape is captured as a graph. So what we do is we try to, the idea of mapper is to try to capture the local properties in the embedding and try to preserve as much detail about the local properties as possible. So in this case, we bin the space, and there are many ways of binning it, and I'll show some of the ways in the afternoon that you can bin the space. So imagine this is the simplest binning is you take the 2D space and bin into overlapping bins, square bins, and then the next trick or hack is, which really helps, and some of the dimensional techniques like UMAP also try to do something similar as well, is you take these bins, and within each bin, whatever points you have, you go to the original space. So you cluster the data within each bin based on the information from the original space. So that's the hack.
Sorry, in the case of fMRI, just so we're clear, are you talking about like a voxel level?
Yeah.
Data is some point, some voxel and some value associated with that. Is that
Great
correct?
question. We'll go to the fMRI.
Okay.
I'm just trying to stay away from
from it because
Okay.
fake MRI, but we'll go there.
Well, the reason I ask is because I get lost if I can't anchor it in,
I'll take you through
you know,
that.
I'm already lost.
So there's a three-dimensional space, just bunch of points, shape is closed to.
And we dropped it to 2D space. That's like a trick of any dimensional reduction to go lower dimension. But the hope is, how do you capture the original shape? And the trick that I'm trying to kind of maybe spending too much time here, but trying to say that this trick really works, which is that you go to each bin, local bin, and try to preserve the original shape in this bin. Don't care about the global shape. Basically, we locally preserve the high dimensional information as accurately as we can. And so, for example, for in this particular bin, green points are farther away and blue and red points are closer, right? In the original space, that's why there's a loop. And that's what you do in the clustering stage. You put the green parts farther away, blue and red stays closer to each other. And then there's the reason we keep these bins overlapping, so that any bin which has a data point sharing between them gets connected by an edge. And that's how you get a graph. And the cool thing about that, so this clustering is like there's a hack piece that you do it in the original space, is that if you look at TSNE PCA versus mapper, you get this graph, and because green is farther away from blue and red are connected, green is not connected to them. So if you just layout, you change the layout of this thing, you will get like a necklace. And basically you'll do this exercise and you'll play with parameters in the afternoon. But this is toy data, like Christina said. Let's go to the brain. But any questions about that part? Yeah.
Is the solution invertible? Like if you go back one slide, that sort of circular representation, can you get back to the 3D?
Completely.
Yeah.
Fully. And that's another beautiful thing, because everybody wants to go back to the brain with fMRI data. Yeah. So we don't lose any information. Everything is preserved. Okay, so then I'm going to show you this application in 4 different domains or aspects of it. So let's start with fMRI data with evoked activity. This is joint work. This is when I was postdoc in Alan's lab, and Gunnar was a mentor, and Gary and Olaf and Peter and Gonzalez gave the data that I'll play with. I will show you the results from. So hopefully this slide will help with the fMRI aspect. And remember this thing, we are into brain dynamics. And I'll tell you, that's why we have time on the as rows. So our features are voxels, right? But if you are into function organization, then you can transpose the matrix and everything still holds. So you can have voxels here and time as your feature, and you can look at relationship between voxels. We are looking at relationship between times. So it's not typical functional connectivity, it's how each TR is similar to every other TR that I have. So it's the landscape of the dynamical evolution of activation. Okay, so same thing as 3-4 or similar-ish. You have 3D in time, so 4D data. I can reorganize it as time TRs by voxel. So in this paper, and this is the original paper in 2018, where we were in the bragging mode. So we had 7 Tesla data at 271,000 voxels. You don't have to go that deep, but it's fine if you want to. And each row is the brain activation pattern at the whole brain level, any TR, right? So we do the same 4 steps. We first reduce the dimensionality from 300,000 to 2. We can go to 3, but the main point that we are going to so low is because we want to visualize the data. You don't have to. You can stay in high dimensional space and still study the properties of the graphs. Here I used a variant of isomap, and again in the afternoon you can play with different versions of this embedding, which obviously makes an impact. And each dot here is again so we have 1,000 TRs. So each dot is again entire row, entire brain activation pattern. We bin the space, simple binning. We just bin it 2D with overlap between bins, and then for each bin you cluster the points based on the original data. So you don't cluster them based on what's in the embedded space, but with the original space. And each dot here, or if like this bin has 3 dots, and each dot is whole brain activation pattern coming from any time. Like there's no limitation that it has to be sequential. In fact, you can shuffle this matrix in time and you get the exact same graph, because you're saying how similar each brain activation pattern is to itself or to all other brain activation patterns across the entire session. Make sense? And then you connect them and get the graph. Yeah.
Just one clarification. So if you get the same solution with shuffling, how does it account for temporal autocorrelation and temporal smoothness?
It still picks up, and I'll show you some results.
Yeah.
Because the thing is, in time, if we have such a high autocorrelation, points, even though you put them down or up, they're going to come back together. In the embedding, they will end up being closer to each other because of similarity. So it won't matter. Good point. Okay, so this is the data we used first. This is from Peter Benetini's lab. Gonzalez Castillo was, I think, he collected this awesome data. So here they have this continuous paradigm. We have 3 minutes of resting state, 3 minutes of working memory, 3 minutes of kind of resting state, because you're like watching Nemo or aquarium and finding Nemo, and then some basic, or maybe very complex math, and you repeat it. And the reason we picked up this particular data set, because it has some kind of ground truth about transitions. So if we are into transitions, so finding the shape is good, but then we really wanted to see how the brain transitions from one state to the next. And we wanted to have some validity. And so if we don't have a transition between these two states, then, it's like, what can you possibly pick up? And also because we have behavior. So we have task performance across these blocks of tasks. We wanted to see whether the shape graph, so the shape graph is the final output of the mapper graph, whether shape graph properties, because it's a graph, you can do all kind of network science on it, from centrality to blah, blah, and say whether there is any property of the graph from degree distribution to whatever, or modularity that relates with the behavior, right? So it becomes useful as well, not just pretty. Okay, so this is one person data. Everything is on a single person level. And this is the kind of spider graph that pops up. And it's gray, maybe a little ugly, so we colored it. And we colored it based on post hoc. So the information of coloring doesn't go into the analysis. What we do is we take each point, each node, and remember each node can be multiple TRs, multiple brain activation patterns, and I know which task they're coming from. So I can color the graph based on which task is where. And that was quite powerful. Again, no averaging was done. So we found that across the 20 or so people we had, pretty much every graph had this core periphery structure. So core is where we found that tasks that required a lot of cognitive effort, so in this case, like working memory or math ended up being. And periphery is where the resting state / watching video kind of ends up being. Because if you think about this, when you're doing a working memory task, I don't know, somehow I'm very bad at it, but I have to focus a lot for 2 back, 3 back, which means that my brain activation patterns are not shifting as much. They're very similar. And that's why they end up being part of the core. Whereas when I'm in resting state, I'm thinking about everything. And you kind of have this kind of, what's the word? I think Olaf had a word for it, excursions from the core.
What's that thing at the top that's not connected?
That's probably head motion. So what happens is if you have TRs and some TRs are very rude, they just end up flying away. But this is also a property of the graph. So meaning I talked about binning and overlap. Imagine I only have one bin. Then everything is connected to everything. It's like the k nearest neighbor kind of thing, right? So there is some magic that goes into picking the parameters. So you can pick up something which is like everything connected to everything, and something which is nothing is connected to nothing. And the solution is somewhere in between. Okay, and again, trying not to average stuff, we represent this data as a pie chart, where the pie chart tells you how many TRs are coming from which task. So it kind of preserves that information as well. Quiz time. And maybe people already know the answer, but one of them is the worst performer on the task. One of them is the best performer on the task. So who thinks left is the best performer on the task? Okay. Who thinks right is the best performer? Just one guy? 2? Okay. So we have 2 winners.
What?
Yes. So why is that? We were surprised too. But so if you, maybe the coloring is not doing justice. Maybe we can shut the lights in the front. Is that possible? Or not? The guy left. But if you look closely, what was that? Thanks so much. The graph on the right is highly modular. It's better, I think. Yeah, thanks. The graph on the right is highly modular. I mean, the red nodes are connected to red, yellow is connected to yellow, and there is like orange is orange. There is high modularity, meaning when that person does this task, they have task-specific brain activation patterns. Whereas this person is likely sleeping inside the scanner, because no matter what they do, their brain activation looks similar, and that's what gets coalesced. Like no matter which task they're doing, either they don't care, probably don't care kind of person. But this property of modularity, it's not that we ran a modularity detection or community detection on it. We said, because we have colored the graph this way, after coloring it, is the quality of modularity different, right? So it's a subtle difference. There's no need to run community detection on it. You can just say, is the quality of modularity based on this coloring separates the graph in obvious pieces. And apparently that what was associated with like reaction time and response time and stuff like that. Okay. So, and then you can go back to dynamics. So the cool part. So you can take the graph again, and this is the invertible part that you were asking. So you can say, okay, if we started with this whole brain across time thing, we got the graph, which is a compressed representation. Now I'm going to go back to time by time and see for each TR that is part of the same node or is connected by an edge, they get a non-zero value in this matrix, right? So they're saying that these TRs are highly similar or some definition of similarity. And you can kind of see the blocky structure pops up from this particular subject. But then what you can do is you can, so for example, resting state is the first one. It's like not really similar to anything else. Working memory may be similar to some things, but also similar to itself. But this case was a funny case because everything is similar to everything in this particular person. But then you can average over, or you can basically treat this as a connectivity matrix, but this is connectivity of, or recurrence matrix of time. How similar each time is to every other time in the entire session for the whole brain activation pattern. And you can say, what's the degree of this graph? And if you compute the degree, you get this. So this is the degree of that graph. And this is average over all 2018 people. And without knowing anything about the task structure, we know that we found the resting state. This is the single TR level, so no averaging, no sliding windows. That you go from like one state to the next. And we ran like a change point detection to pick up these 8 states belonging to 8 blocks, which tell you, apparently the similarity here tells you the cognitive effort that the task needs. So it kind of encodes that as well. Cool. And then this is the tool I'll talk about, dinosaur. Hopefully it never get extinct, but so this like, that thing was just for coolness, nothing to do with neuroscience or science. And then we'll just hit the play button. And then what I'll do is it'll basically highlight nodes where that TR is. So this, you see this continuity is kind of captured because the nodes, time points which are highly similar to each other end up kind of staying in the same place. So this resting state is kind of traversing the resting state space. You can get activation, deactivation, simple spatial mixture modeling. This is like significantly activated, deactivated, single TR level. And now this going to the memory task, so I spend more time in the core, and then it'll jump out eventually to something else. You can recolor this graph by things like hit, miss, blah, blah, whatever. So I think I do it at some point here. So I said, okay, I'm going to recolor the graph and look at where the hit, miss, or wrong happens, like in different tasks. And you can see whether whatever task you're into, maybe the hits happen on the excursions or miss happens on the excursions. Also. But this ability of getting activation, deactivation significant, this is like cluster corrected at the single TR level is possible because at each TR, I'm not only looking at one TR, I'm looking at all the TRs that are connected. That gives me this lens to reduce the noise and increase the signal to noise ratio based on this embedding, which otherwise is very hard to get.
So do you have to do any kind of different pre-processing on this?
Nothing.
Because you've got task rest. You do any like low pass filter or band pass?
So in this case, if I remember right, we did the standard CPAC. Those days was CPAC was a fun thing, but CPAC processing with, I did do a temporal pass, but I made sure that temporal pass was not affecting the fastest event that we had.
So like 0.01 to 0.1.
Yeah, something like that, yeah.
Standard.
It was a slow block design, it was nothing fast, but you can change it according to your time. Yeah.
So the point of the original Gonzalez-Castillo paper, these data are from, is that these 5 different sort of states have very unique connectivity signatures, so unique that they could be decodable with nearly 100% accuracy, like on individual blocks.
Yeah.
So sort of the idea of the brain having sort of different patterns for the states, I guess I'm wondering like how does this sort of expand upon that? Maybe that's where you're going with that. Like you could just look at a state by state similarity matrix, either of the spatial pattern of bold or the connectivity pattern, and see that some states like maybe resting is more similar to video watching than it is to math, for example, and see that for each subject. So sort of when you're looking at these graphs, you get like a lot of fine structure, but how to make sense of it, I guess.
That's a very good point. So you're right, in Castillo's paper, that's what they were trying to do. And I forgot whether a single person level or group level, but they were trying to use functional connectivity to sliding window or something to separate them.
Yeah, I mean, they showed data within each subject and within each block, like at the level of, I don't know,
Yeah.
20 or 40, 20
Yeah.
to 40-second segments of data. You could tell from the connectivity pattern which of the 5 tasks.
Yeah, good point, good point. I mean, we could potentially do some kind of clustering here and maybe separate the tasks too, but I didn't want to do that because here I wanted to see the transition between tasks. And if you're doing a working memory and math, the idea that they will be separate in your brain doesn't make any sense. There has to be some overlap, right? Like for example, in subjects who are like perfect, like that guy on the side, yes, the red dots are kind of separated from the yellow dots and stuff, but there still has to be some transition, some overlap. So we didn't go for that question, but you could potentially, do some clustering on top of this, or run a community section on it without the coloring and see whether, oh, I can perfectly separate the tasks if that's what you want. Yeah, we kind of stayed away from it. The other reason staying away from it was that functional connectivity being as awesome as it is in code and code, but the task had different trials, right? So its math was like slower than working memory, and then video, you press a button every few seconds. So just button press can differentiate within the task. So, but nothing against that idea. It's a good idea, but we didn't go for it. Yeah. Any other? Okay, and then, oh my God, half an hour into it. Then let's maybe go a little bit faster. This is the other paper where we looked at, instead of looking at evoked activity, let's look at resting state where there is no task per se. And this is joint work with, this is Nico's data, the Midnight Scan Club data, and Damien Fair and Raphael, who's apparently now an astronaut, which is amazing. Yeah, I don't know what we did to him. And then Max Shine from Australia. Okay, so rest is far from restful. I guess you all know that.
Wait, I want to see that.
Oh, sorry. Okay. Yeah. So you guys do that too? Good. Cool. Okay, so rest being rest, how do we create a landscape of your resting activity? Meaning in task, yes, extrinsically, I'm making you go from this task to next task, blah, blah, but in resting state, you're doing that to yourself. So, but how, is there any rule that govern your transitions, brain activity from one state to the next? Do you go directly from one state to the next? Like if this one state is default mode positive versus frontal parietal positive, do you just jump, or is there a transition state that you have to go through? And whether there are individual differences, that's what we were into. These are 3 questions we wanted to ask. And the data we used is Midnight Scan Club data, as I mentioned earlier, 5 hours of resting state, individualized parcellations, and then we applied Mapper to it, and then we had some split data validations. So the way we did it, because we have 5 hours of resting state, we split it into 2.5 hours, 2.5 hours, and we showed whatever we find in the, I think in this case, they had like 12 sessions or 10 sessions of 10 weeks of, so we split it to odd weeks and even weeks. So we get 2.5 hour week worth of session, and we showed that whatever we find in one session replicates in the next session at a single person level. We also use some null models, which are also very important, by the way, sidebar for any dynamical analysis. So we use AR and PR, phase randomized and autoregressive, multivariate, and then also replicated this in human connectome data. Okay, so even session, odd session, take the pick, and that gives us plenty of points, individualized parcels. Again, this is time on the rows, and you get this kind of very dense mapper because the number of points go up. Now we have 5,000 points versus like 1,000 points. But the idea is the same. Each node has multiple TR. They could be coming from anywhere as long as there's similar definition of some similarity. Size of the node tells you if they had more TR, less TR, whatever. But now the problem is, how do you color it? So there are many ways. One way is you just color it based on the topology of the graph itself. You can say, color the nodes highest who have the highest degree versus like something else. Or just look at the degree distribution of this density, this plot as well. And we found across all people and sessions, we found this fat tail, kind of very obvious across people. So we ran a comparison against the null models, and indeed there is this fat tail. And this is odd session, even session, 10 subjects twice. And we see this big, dense fat tail, which means that in this manifold of activity at the resting state level, there is some states nodes which are have high centrality and have high degree connectivity, meaning they act as some kind of transition. They're hub states, so to speak. Again, but in time, we're not finding hub in the brain like default mode PCC. There's hub state at the whole brain level in time. And you can then obviously color it, find it, where they are, how do they look like, and I'll get back to them because they have very interesting properties. The other way of coloring the graph is you can color it based on, we have individualized network definition. So I can just say, hey, color each node based on each of these resting state networks. And again, we use pie charts. So like if you have 100 TRs, how many of those TRs we z-score the resting state activity itself? So we say how many TRs go 1 standard deviation above activation in default mode network, and 1 standard deviation above in like frontoparietal network and stuff like that. So you take some threshold and you simply, just for the coloring part, that's the only thing we're doing thresholding for. Everything else is in as is. But just to color the graph, we say which nodes have 1 standard deviation above or 0.5 standard deviation above activation in default mode versus ventral tension versus blah, blah. And that gives you this kind of pretty-ish plots where if you think of this as a map of US, then Texas get default mode network, California get frontoparietal, makes sense, and other regions, right? Sorry, bad jokes. That's why I didn't want you to record this. So, and then the same person, so it's like odd sessions and even sessions, the graph doesn't look exactly same because it's like the central limit theorem. Eventually, when we have all the TR, like hours and hours resting state, maybe the graph would approach some kind of similarity. But the cool thing is this, which networks hang together is similar between people. So like default mode likes to hang with the blue guy, which is I think ventral tension, frontoparietal and dorsal tension likes to hang together in this guy, which also hangs in the separate session and stuff like that. So this idea that, you know, like if you do hidden Markov model or some other model, you have to pick like one network that goes up and you have mutual exclusivity and stuff. You don't have to do that here. You can really see individual differences, not just in the landscape, but also which networks go up and down together. And there is no limit. You can have 3 networks going up and down together for each different person.
You've got a question?
Sorry, yeah.
I'm curious how you make sense of anti-correlation between networks based on these graphs, because it seems to tell you a lot about what does hang together, and you can kind of infer what doesn't hang together. But is there like a clearer way to come to that?
Yeah, love this question. Yes, we did, and we found it was like just the reverse. So what we did in this case is I'm only looking at positive. So each node is colored by positive activation beyond 1 standard deviation or whatever. But if you do the other way around, you look at negative, then this picture reverses. The default is negative here rather than positive here. So it's like it reverses itself. But we can go deeper in that. We didn't look at like absolute or some kind of phase blocking or anything like that yet. But that's a very good point. Good. Okay, so when we get this manifold, you can obviously check it. You can take each node, any node you want. In this case, I didn't even do spatial mixture modeling. I took that node, just averaged TRs and put it on the cortex. By the way, everything is cortical activity only. There's no subcortical data because it's not clean enough, apparently. And then this also shows you for each TR, like default mode is above this band of 0.5 standard deviation. You can look at frontoparietal, single opercular, same way. Frontoparietal was kind of cool because like apparently Dan, frontoparietal and salience like to hang together in this particular subject. And this is the negative part that you were asking. So the other one goes down. You look at them as well. But hub was the coolest, like this uniform rainbow. Apparently Midwest wins, but anyway, where the equal representation more or less of all networks. And as I said earlier, you can see which networks hang together. So this is a way of capturing that. You just look at on the manifold, which networks go up and down together. They're highly subject specific. These are 2 different subjects across 2 different sessions. You can kind of do this fancy matrix, people by people, and show that within participant, there's high subject specificity of which networks hang together. You can again go back to time. So we took these manifolds and brought them in time, meaning each node, here we had to take winner take all, otherwise it would be multiple bands. So we said for each node, whichever network is highest winner, all the TRs in that node gets the same, or like a weighted label. And then the gray ones are the hub nodes, which are in the center. And so you get these beautiful bands. These bands also somehow look like army bands, but you have this MSC, like the different subjects have similar networks that they go through together. And then easiest thing to do is you just do a Markov chain on it and say, what's the transition probabilities go from one network to the other? You get these source destination things. Apparently hub states, this is just one way of showing it in time as well. So we show that hub is central in the graph, but if you look at the initial time points, TR hub states are the most significant or sought after. From any state, you have to go through the hub state apparently. And these Markov chains, so hub is again kind of shown as a high centrality. And these Markov chains are also subject specific, more so than between persons. So cool sidebar, now I'm working with Emily, Finn, and other people who are doing this on naturalistic data. Happy to chat about that as well. And we're finding that the kind of movie you watch beautifully captures which kind of states you will go into through this kind of approach. So, okay, so for resting state, we find that yes, there are some rules. You're governed by this manifold where it's not like randomly you go from one state to the next. You have to go through the hub states, and then there are individual differences in which states co-occur together and so on. So, any questions? Yeah.
I just missed, like, what is the hub state? Is that a set of brain activity that's visualizable? Like,
Yeah.
does everyone have the same?
Great question. Yeah, so we tried to visualize it, and it was like mean activity across the entire brain. It's like it's one of those states where like nothing fun is happening.
So do we know it's a hub state, or the brain might be just changing from one dominant network to another on a fast time scale, and fMRI basically just smooths it so that during those transitions, it seems like a wash.
Fake MRI, anything is possible. But the other thing,
To assume that the brain always has sort of one network, it's just, and any TR one network is the dominant network, and then the next TR is either the same or it switches, but all these networks are working together, so like.
That's a very good point.
and so we tried to look at hub from like just putting it on the brain. We found like mean activity, nothing popping up. Then we haven't looked at subcortical yet, but another grad student who was visiting a student in my lab, he tried to look at through PET images. So he tried to map it on PET data, and he found that, and that paper is kind of coming out now. They found that it's the lowest metabolic state at least, but that's all we know.
Okay, thanks.
Yeah, Tashia, yeah.
Well, I was just, a stupid question, but do you run global signal regression on this before or you don't?
Oh, global generation.
Because you talked about the mean being the hub.
Yeah.
Yes, we do.
You do?
think so. Yeah, this is Nico's lab. I mean,
Oh,
come
yeah,
on,
of
they will
course.
kill me if I
Yeah.
again cut it out.
Oh,
Um,
okay, interesting.
Yes. Yeah.
I have a question from earlier, I guess. How much does the ultimate graph depend on the dimensionality reduction method you use?
A lot.
Yeah, because that's is the key. So for example, so when we, and I can check it out in the afternoon session as well, if your dimensionality reduction is giving you a big blob and everything is connected to it, like next to everything, then it's not going to pick up. So it's a lot of things matter. So we have another paper coming out. It's in bioRxiv now, which talks about choices at each step of mapper. So like what kind of distance you should use. The Euclidean apparently works best. Correlation not so much. What kind of should you use? And another kind of binning that I didn't talk about here, we'll talk about in the afternoon. Should you actually, we have a newer mapper version where we do not do any dimensional reduction. We stay in the high dimensional space. That's called intrinsic binning, but it has some of its parameters on its own. So everything has its parameters and it depends. Yeah.
Similar to that, what kind is there a particular choice of clustering that matters?
We looked at DB scan and like linkage, single linkage, blah, blah. And apparently single linkage is as good as anything else, at least for the partial clustering step.
So you usually don't do KV2, more density based.
Yeah, more density based for partial clustering. But again, all these are awesome questions and, fun papers potentially to try out different things. Yeah. Awesome. Okay, so this is going to the mental health disorder stuff. So I am, again, lucky to get some more data from Damien, Leanne. Damien is giving some, he is working on his ADHD data set and ABCD data set. Leanne is providing some of the depression and development is coming from her. And again, depression from Ian Gottlieb on this data set. I probably won't go into too much details because I don't have full results yet. That's another reason. But I'll show you results from the ABCD data set that we have been playing with for a while. And this is not published yet, not peer reviewed. Don't take my word for it. So in this data set, as you may already know, you have like 20 minutes or so of resting state per person, per kid. And we only looked at baseline data. And we create the similar approach. And here they had some kind of
Are you concerned at all that it's 4 or 5 minute blocks and you're pulling them together? That
it didn't, yeah.
it doesn't matter?
It doesn't matter to us because again, we can shuffle time and
Okay,
we can pick up that.
so that you don't need 20 continuous minutes.
I think it's better.
As long as it's less motion.
Um,
Apparently people need to move. And so we create these graphs, but then they are not as high dense because now we are down to from like hours and hours of resting to 20 minutes. By the way, that's a big problem with Mapper. I'll just say it out loud. Like if you don't have enough data, you're not going to get anything useful. So if you have 5-6 minutes worth of data, it's probably not a good idea to run mapper on.
Is there like a baseline that you would set as a good bar to clear for each number?
Yeah, it's like you can try it and see if the graphs make sense, maybe. Because if you have a very high TR, like a high resolution, then maybe 5 minutes is more than enough. So you can get like, I don't know, 100 points or something. So it depends on how many data points you have and whether they can answer the question. So we applied it to resting state on ABCD. The graph doesn't look as cool, but you can do the same thing. You can create like this kind of co-occurrence matrix and then do some kind of stuff on it. What we did instead, and this is something maybe you guys can try, and this is incoming, so not peer reviewed, don't trust it fully, but we developed a relatively newer method, which is population mapper. We're calling it deep mapper because in Silicon Valley, everything has to be deep now. So we take participants, however many, so you run mapper once, and then the input to the next mapper becomes a mapper. It's like a hierarchical mapper, layered mapper. And the idea with this is that, so now instead of having nodes being time points, now the nodes are people. So people whose graph properties are similar end up being part of this bigger population graph. And kind of helps a lot because now I can color the thing with who has ADHD, light up, stand up, kind of thing, right? So like you can see where they are. Okay, so this is how it looks, and I can color it. The first thing I colored it with just the proportion of hub nodes. Remember the hub nodes, resting state? So turns out, although we didn't give hub nodes as input in particular or separately, this mapper kind of picked it up, and yeah, I love Fibonacci. And you get this low proportion of hub nodes and kind of goes all the way to like highest hub nodes. And here's some examples. So like I'm highlighting the hub nodes in those graphs, less to more. It's like the proportion, so ratio of hub nodes go up. And then just for fun, this is happy to talk more about if you guys have ideas about how to analyze it, because we're kind of a little bit struggling with it. But simplest thing I did is I just did a median split and said, okay, color the people who have less median hub proportion versus more median hub proportion. And there's not like perfectly separated, like they're kind of all over the place. And then we looked at, like Damien had, some of his people have done this where they have 3 main PCs, like general ability, executive functioning, learning memory, and then emotion dysregulation. And we found that, and this is where I kind of like population stats, but you found that the people who have less hubs are apparently better at executive functioning and learning and memory, and emotion dysregulation is the other way around. So it's better regulating emotion as well. So more hubs is apparently bad in this data set. This is baseline ABCD. Then what we did is because this is like general population data set, so some of these kids actually develop or get diagnosed by ADHD and other disorders. So you can just say, okay, highlight the nodes which has any person with ADHD. And again, we found that it's not like one ADHD, it's like all over the place. That's what people like to say these days, that ADHD is not one monolith thing, it's like all over. But then you can do the same thing, you can probably do many things, but one thing we did again is like, said that people who have median high hubness or low hubness versus high hubness, and then look at the 2 ADHD. So apparently like symptom severity goes up if you have more hubs. So kids who have more hubs and have ADHD have more symptom severity. So some link that we're trying to make with ADHD. But that's all I have about. Any questions about that part? We're still working on it. So any idea?
So especially with something like ABCD that's longitudinal and part of its aims are to think about outcomes across development, how might you sort of leverage a tool like this to look at like property change across time?
Yeah, that's exactly the hope that we could do it. And so there are multiple ways you can think of doing it. The cool thing about Mapper is you get a graph. When you get a graph, you can think of like having another graph, and you can do multi-layer community reflection and all kinds of fancy things like saying, hey, the node was here, did it move to another location, did it change, did it switch? Because it's a graph, you can do all kind of fancy network science on it. That's the whole. Any other question?
I was wondering if in case the hubs that we were looking at, maybe we can further categorize them. That will help us understand how much connection they have within a community and then between. And possibly it's not just the proportion of the hubs, but maybe they're switching their role. And that is possibly helping with executive functioning. For example, in executive functioning, we would need some of the brain areas to switch, for example, to a peripheral non-hub in order to communicate with another network or community.
So,
Not just number of hubs, but the location of hubs on the manifold itself. Maybe who is next to them is more important than just having X number of hubs. That's a very good point. Yeah.
So this idea of running mapper on the graphs of the individual subjects, mapper data, and producing that sort of representation of subjects, it's interesting, but it's very abstract to me. Like, how would if you just analyze the hubness of each person and then do those t-tests like you were showing, do you get the same thing? Like, what do you gain by putting all the subjects into this graph that we're looking at? Like, is this?
so for example,
What we got was, so we think of mapper as, it is purely exploratory data analysis, in other words, fishing. Meaning, I had no idea hub would turn out to be this case.
No.
I had no idea that hub would actually be a cool property to look at.
Okay.
So, or it will matter for ADHD or not. And so whatever I'm showing you, so we also did a split analysis. So it's like 11,000 kids split into two similar things. And apparently the clean data from Damien's definition of cleanness, we only have like 50% data clean. So like 2,500 kids is this graph, and I show the exact same replication of 2,500 kids in another graph and stuff like that. And then we show the same thing on an ADHD enriched data set that it matters. So this is more of an exploratory data analysis technique to help you see what could be interesting. Like when I did the first paper, I had no idea that separation of task would actually relate with behavior. In that case, I'm guilty of fishing, yes. But we do then replicate it. Okay, last thing, I'll go quickly over this. So we are also looking at being at Stanford is cool because you get these cool people there who are actually collecting real data. And I don't have to collect it myself. So they have these pre-post interventions. So Monique is actually an MH. I will show some published data that we did with her. She gave people Ritalin or methylphenidate and see how that improves their working memory. And we found that Mapper picked up very cool signatures of those improvement in the brain dynamics. Robin, you probably know, he's the psilocybin stuff, and we are getting his data and looking at how psilocybin changes your brain dynamics. Nolan does a lot of TMS, and Jessica does like ADHD medication with methylphenidate as well. So we have results on all of them. So happy to present them later. But this is one of the cool papers from Monique. Monique's data set where she gave people a threat of shock paradigm, and within that paradigm, she also so they were supposed to do this working memory task, 3 back, 1 back. But right before the task, they gave them methylphenidate. Methylphenidate, if you don't know, it's like Ritalin. It's supposed to apparently enhance executive functioning. And they do find that people who take MPH, their behavioral performance is much better than people who are not on MPH. And the question was, how is that happening? So what we did is we looked at, again, mapper graphs. This is during working memory, like 2 or 3 runs of working memory task. And this is showing you one person, another person. This one is MPH, this one is placebo. And across people with MPH, we found this nice separation between the condition. The condition which did not require too much effort was nicely separated in activation patterns than condition which required a lot of effort. And we found that this actually helped people, or methylphenidate somehow helped the brain use the resources efficiently. Meaning you don't need the same kind of resources to do 1 back as you need for 3 back. So we have to still replicate this as well. But the idea is that this drug is somehow efficiently allocating resources or making or facilitating that. And we could see that in Mapper, which you can't see in like GLM result or something like that. You can quantify it using some kind of participation coefficient. Everything is like high value, low value is when they're segregated. And we found MPH was more segregated across task conditions, and that was also related to behavior improvement or behavior. Okay, cool. Future work. Almost at the end. We'll talk about some methodological advances that we are doing, and we'll talk about this thingy dinosaur. The idea of dinosaur is, so I didn't tell you this, but TDA is a very big community, math community, and there are many people developing these map algorithms, not just us, but we are trying to kind of create this, or we tried using dinosaur, we tried to create this extra layer for neuroimagers or neuroscientists that can use these fancy mapper tools or other TDA tools and kind of put it in the brain aspect. So I'll share this tool with you. Credit goes to Caleb. I'm not a Python person at all, so I'll probably mess up big time today. But he created these nice notebooks that you can play with. I have some similar algorithm in MATLAB, which I'll also share. Caleb, again, and Samir, a postdoc in my lab, came up with a new approach called new mapper, a twist on new. And the idea is like, it's highly scalable. So if you're into computer science, it's like the big O is like constant, meaning as compared to other mapper that exists. And as I mentioned, there are many other mapper tools that exist in the math space. So the cool thing about this new mapper is that not only it's scalable, can deal with millions of rows if you have, we don't deal with that kind of problem. But if you have like genomics data and other data, you have billions of rows, it can do a very good job scaling. But also it does not require any embedding. So the first step, going down to low dimensional space, they took it out. So they do this, and I'll talk more in detail in the afternoon, but they do this by creating a k-nearest neighbor graph in the original space and then doing binning on the graph. So rather than splitting the data into these bins that we need in low dimensional embedding, you basically consider imagining for each node in the k-nearest neighbor graph, just blowing it up. That the radius is your binning. That's what it is. That requires some fun farthest point sampling stuff that they did, and I'll talk about it more later. And also, I don't know if you're familiar with this, but in deep learning, this thing called optimal transport is very common these days, and they use that to also infer the brain dynamics. Again, more for afternoon. Mungsen and Samir. Mungsen was another postdoc in my lab. Now she's an assistant professor, I think MSU. She came up with this very cool idea that, you know, we have this mapper, but it's not directed. So why not make it directed? Because we know which time is where. So you get this directed and becomes an asymmetric graph, which is really useful. And she showed that using virtual brain kind of biophysical modeling, that we can, in the virtual brain or the biophysical model, she simulated transitions in arousal, like global coupling, and showed that temporal mapper picked it exactly, as compared to dynamical functional activity analysis could not pick it up. That was a very cool stuff. So everything I'm sharing is like on GitHub, lab's website, you can play with it. And DMapper, Daniel is a software engineer, now a grad student. He, as I mentioned earlier, he took each state of mapper and kind of showed how changing different variables affects the graph. Okay, so to end, I only had time to talk about the visualizing the dynamical features, but I think to solve this problem, we also need biophysical network modeling, some kind of real world experiments, and validating the nosology. And thank you so much for having me. These are the people who are thankfully giving us money, and the awesome lab members and collaborators, especially Lucina here and other people who have helped with these projects. Thank you so much. Any questions?
Well, you've given us a lot to think about over the lunch, so hopefully everyone stick around, grab some food, and bombard Manish with questions while he tries to eat. And then we'll also have the workshop in the afternoon, which is hands-on. So, I mean, this is really mind-blowing, and I think it will take us a few minutes to process.
Maybe I just showed you things that work. And there's behind
Ice cap or whatever they call it. Right? There's plenty of things that don't work.
We'll have plenty of time to get into that. So yeah, feel free to grab lunch. Thanks so much, Manish. There he's gonna go through some people who want.
For mapper?
For this dinosaur.
Wow.
Okay, let's like figure out.
Daniel.
this because it's-
Let's see this.
Okay, so what-
Down from Paris.
Oh yeah, I saw Paul Thompson. Pictures.
Oh,
from-
okay.
Yeah.
Yeah, we were talking and you eating lunch.
Oh, yeah.
Yeah. Yeah, I was like, when I saw it, I was like, oh, I should get on
I should
there.
have gone.
I should have get on, I'm not on like the USC mailing list, so I.
Well, it was the last minute thing.
Oh, yeah, last minute.
Dara, have you met Daniel?
Hi.
Daniel Arkin?
I've seen you a lot.
Yeah.
Most people could show up, but I feel like when I meet the person, nobody ever comes today.
Oh, well done.
Okay.
Yeah.
Yeah.
So that's why there's
We
like.
recorded it, so.
Fantastic stuff.
It's undersold. It's so cool.
Yes, it
Like
is.
nobody knows. And I'm always trying
Yeah,
to.
you should,
Podcast his, like, use this dinosaur. I don't even know how it works.
Yeah.
It just seems
Great
like,
point, yeah.
anyway, he's around. Or until 2.
We had a nice chat. It reminded me about how. We had a nice chat at residency.
Oh, you did, okay, so, well, I mean, yeah, there is lunch, there's a whole.
It's very nice. It just, sorry, it took me.
Oh, yeah.
It took me a very long time to figure out how to start.
Yeah,
This is-
I'm very bad at getting people here, I'm very apologies.
No, it's
I
like,
literally was in a Italian dissertation committee at 7:30.
in Italian,
It was, but you know, like I was zooming into.
for who's dissertation?
You know, Michael Lombardo, who works in autism. Very nice. Anyway, I was busy all day.
When
but
do you head off of?
Tomorrow morning. However,
That's a big pile of bullshit.
well, first of all, how was the practice?
It was lovely.
Okay, good.
It was lovely. It was like a bunch of artists. I was the joking scientist.
Artist?
You can just do something like this directly.
But
I forget why I needed it, but I still haven't removed it from my bag.
And I would like sit
It was
there
all
for
probably.
so long, but I never caught anything, yeah. But there,
I remember like
some people catching, some
Just
people
for a
are catching.
looked forward to having a check because my mom was one of the people that
It
would
was poisonous.
take out time in the grocery line to like
No, I've never had.
had like okay, it was $55 for groceries like I'm ready and I just thought that was
In a while, but
I don't know why, but I enjoyed observing that process and I really looked forward to it. I like have had like outdoors check out ever since, you know, like everyone
Yeah.
just uses debit card. Like, okay, 3 situations maybe in your life where you like it's like security deposit
We catch.
and whatever else weird. The next time I move, I'll need another one.
Straight. These ones get really big, the straight. But these are like the common fish we would catch. And then we would also catch, you couldn't eat these, but they're really good. We'd catch a ton of these, called sea runs. And they like have like these weird little forky things.
Yeah.
Give me a second, just need to put something in the back.
Really search my way to ocean, kind of taste like the saltwater.
Yeah.
How was your class? Oh, it was great. I've seen,
But like you just crack them to this point.
I won't list them if I can.
It's like, it looks like orange.
There's like
It's very productive.
Exactly.
the pond
Oh,
that's
I
very
think I've seen it
kind of
before.
wet.
This is what it looks like.
Eastern West Hollywood. And there was
This is what it looks like.
so I was like in the general admission like reading section. There was like one perfect right in the middle. Just kind of going through one right in the middle seat.
You should do your session.
Everyone's sitting on some milk.
Yeah, so it's not like a.
Sat there, it was fantastic.
Yeah.
No, that's fair.
But I respect that you had to.
I mean, it's safe, I know, take care of it.
want to type that in, you can go ahead and download the package.
You have to do a pip install. So these are in Python, which should be pretty straightforward. You install dinosaur with that spelling. In Python means Python, of course, something won't work. But then I have another tutorial in MATLAB.
Okay, can you hear me okay now? Better. Oh, should we start? Huh?
What was that link?
Oh, sorry. Oops, there you go. So github.com/brain dynamics lab/dinosaur, DYNUNEUSR. And this first link already have the link to these notebooks as well. So if you just go to the first one, it should be fine. So that's the dinosaur thingy.
We just run the setup.
You can just do a pip install. Or you can download it and we can just go to the notebooks as well. Now I'm lost myself. Wait a second. We didn't have installation page. I thought we had one. Where'd it go? Yeah, so if we just do pip install dinosaur, that's all you need to do. Oops, I'm not showing you that, sorry. So this is all you should need to run, and it should be fine.
3.2 Python.
Which Python you mean? 3.6? And then there are notebooks as well. So let me very briefly talk about it, just in case you missed the first part of the talk. Brief introduction to TDA, and then I'll go straight to. Okay, cool. And so we talked a lot about, oops. We talked basically much about the mapper part, where you take whatever the data is, you bin it in some way, and then create the shape graph, originally made by Gurjeet Singh in 2007. But there's another very cool method called precision homology of TDA as well, which is also a lot of fun. And the idea is kind of similar, that you have a bunch of points and you want to create a shape, or you want to learn the shape of this object. And the difference between mapper and precision homology is it has this idea of persistence. So I mean, maybe stop the movie and like. Okay. So again, just like mapper, you have this bunch of dots and you want to create a shape out of it. So I'm just giving a very brief introduction to precision homology, and I'll shut up about it and we'll go straight to mapper. But the idea of precision homology is, do you guys know about this by any chance? Any one of you? Okay, so let me try then. If you have a bunch of dots and you want to know the shape of this object, just like mapper, we want to know whether this is a closed loop or not. And the way it is done is slightly different than mapper. So in this case, you take each point and you literally start blowing it up. So there is a variable called epsilon, which is the radius around this dot. And as you keep on expanding that dot, hopefully you can see the animations, the minute it intersects with another radii, another circle, you create an edge between those 2 points. And you kind of keep doing it until the radius is too big and everything is connected to everything. And so the idea is that in these barcodes here, you're measuring what kind of component appears. So at this point, if I go back in the movie, here, the first component or a number of the y-axis on the bar is telling you it's like different level of components, like one connected component, two components, or a loop pops up and so on and so forth. So at this point, two components are existing and they will live for a life. And how long, like this x-axis is the diameter of the balloon or the ball that we're expanding. And at a certain point, everything, these two components that were here, they disappeared and only one component is alive. And that's like the whole loop. And that big component kind of stays alive for the longest possible duration of the radii. Right, so you keep expanding the radii, and at one point this will also die. So that component disappeared. So the idea of persistent homology is how persistent is the shape across the value. So this is something with Giovanni Petri and others we are collaborating with from is in London now. The idea of to use this so that we don't even have to worry about fMRI-based thresholding. You can basically go look at your relationship between points across all thresholds and find out which is the most persistent component. And that gives you a much better handle on the shape. So this is like a precursor to future work, hopefully. Okay, sorry for the digression. Going back to Mapper, we already talked about this. The idea is we will go from 3D to a 2D object while preserving the shape. And this partial clustering is the fun step. But today in the tutorial, we'll start with the trefoil knot again, like the synthetic data set, and also some fMRI data set. But with the synthetic data set, you can feel free to try different embeddings and different binning functions and see whether you get a handle of how parameter selection affects creating the shape. And the hope is that, as we said earlier in the talk, they will get the graph which will capture the shape. Okay, sorry, this is the link. So hopefully you guys are on the link. So we'll start with going to the Dinosaur website and then doing the pip install. Hopefully it's done for some of you at least. It works, yeah. And then go to the next link, or it's in the same lab GitHub called Dinosaur Notebooks. So it uses, we'll basically do a Jupyter notebook, but it uses this, this is a logo of another mapper tool called K-Mapper or Kepler Mapper, which is very famous. Mapper tool, as I mentioned earlier, in TDA community, all of these tools exist. And so Dinosaur is kind of hopefully help us out, kind of use fancy tools, but for neuroimaging in particular. Okay, so now we'll switch to, so if once you install the Dinosaur, and let's go to the notebook.
So were you able to get it installed? I tried to put in the git clone and then it gives me an error.
So did you just try pip install Dinosaur? Did you try that?
Just that one?
Yeah, that's it. So don't do anything else. Because I won't be able to help with anything. Just do pip.
Python, what am I doing?
No, like on your terminal, you have pip installed, and you just do pip install. If you do like a brew something, but or wait for the MATLAB. I'll show you the MATLAB. If you have MATLAB, you don't like Python, I have the MATLAB version too. Okay, so can you guys see the notebooks? The link works. Okay, so let's try the first one, the trefoil knot. And I'll open this on my machine. So hard to work with. So, okay, good. So this is how it should look like. And with any Python thing, we'll just go through each of these cells. We're just using NumPy and some SciPy and some Network X libraries, just for Matplotlib for plotting, nothing as crazy there. And then Kepler Mapper is the mapper function we will be using that will be then visualized using Dinosaur. So you can get it in, and it kind of is very similar to your typical PCA T-SNE. How you run PCA and T-SNE, like scikit-learn, you can use the same way. Dinosaur you can import as DS in this case. It has already built-in data sets like the one we will use, trefoil, and you can import some of these functions as well. I mean, this is very self-explanatory, I guess. So I'll keep going. First, let's make the trefoil knot. And if you just plot it, it is supposed to have these 3 X, Y, and Z variables. If you plot them in, there's no time really, but across samples, it's supposed to look something like this. Like, remember the trefoil knot, the closed loop? You can visualize them separately. And then this is basically giving you a different viewpoint. So X, Y, and Z versus you can look from X and Z and Y is coming out of you, just looking at different angles, just visualizing it, like camera orientation, basically. And then let's do the dimensionality reduction. So the first one is like PCA. When you do PCA on that 3D structure, you get something like this. So you lost the ability that it's a loop. I see heads down, so I don't know where people are, but I'm hoping you're going through it, or maybe go through it yourself. It's either way is fine with me. But let me know if you get stuck in any place. Similarly, T-SNE, you can apply T-SNE and you get like a point cloud, which doesn't look like a loop, but it's just a bunch of points because you lost the third dimension. And then let's do the same thing with mapper. Here, because it's such a simple data set, we're just projecting into like X and Y, and from 3D 0, 1, 2, you're just going to 0 and 1, just first 2 dimensions. I'm using dbScan, but you can try anything else you like. And cover is the main mapper kind of variable or function that takes resolution, which is like how many bins you want. So remember in the, or maybe I can just show you in this example, when we talked about binning the data, the resolution tells you how many bins you should have. So if I say 6, then you're going to have 6 times 6, 36 bins. And the overlap tells you it's like 0 to 1. So no overlap is 0, 1 is 100% overlap, and you just go in between. Yeah.
Just a question. For the dbScan, I see you picked an epsilon of 1. Do you just always use that?
No, just this for this. You have to play with these parameters. So for the trefoil knot, I just use 1, but feel free to use anything else.
Okay.
Yeah. So one thing I didn't talk about, actually, I should have probably talked about, is let me bring that up. It's an important point. That whenever we publish these stuff, just like any good reviewer would ask about, hey, did you look at different parameters and how does the result look like? And so to do that, we typically present this graph that I'm going to show you. Yeah. So example, the 2 main parameters are resolution and bin. So these are the figures I showed you in the talk earlier. So I'm just showing you the plots at different parameters. So if you have least amount of overlap, for example, or highest amount of overlap, sorry, and then least amount of resolution, you still get everything disconnected. So the graph that looks like the graph I was showing in the talk are somewhere there, but as you can imagine, if you have lowest resolution, meaning only few bins, then everything gets connected to everything, versus the other way around where nothing is connected to everything. And there's this big patch. So whenever you show, so here I'm showing you that, I can barely read myself. Okay, this is a correlation with behavior. So here I'm showing you that among all these parameter values, there was this big channel, which makes sense, that kind of exists here, which has that property that you're presenting preserved. Meaning it's not just that by fluke, one parameter choice gave you the result you wanted and everything else doesn't work, right? So we almost always show this kind of a parameter perturbation analysis to show that the results are not dependent on just one parameter combination. And that's what you should do too when you use mapper, because it's possible that just by chance, one parameter has the best hypothesis that you may have, but it may get disappeared when you use a different parameter. Okay, so here, 50% overlap, 6 bins times 6, so 36 bins. And then just like scikit-learn, if you're familiar with scikit and other machine learning in Python library. You just do a fit transform. X is your data. Projection is projection, which we just use like first two dimensions. And then you just map it. Lens is the lens we made, which is this first lens. And then X inverse cluster, so whatever cluster you use, I think in this case, use DB scan, and cover is the cover we define here. And then it spits out the mapped function or values. So it basically says that, as I said, you have 6 bins, then you're going to get 6 times 6. 6 is the resolution parameter. You're going to get 6 times 6 bins, which we call hypercubes, because this doesn't have to be in 1 or 2 dimensions. It could be any number of dimensions. You have 36 hypercubes, and based on the data, it made like 69 edges, blah blah, and apparently 38 nodes and stuff like that. And then the cool part comes is you can actually visualize these things. So this is the filter function itself. This is your basic T-SNE or PCA, nothing fun, but you can then create a shape graph out of it. And it spits out the graph. You can also visualize intermediate stages, like if you want to present it to the reviewer or whatever. Typically, the data is never as clean, but it's still fun to look at. This is also where you can use different parameters. So for example, if your data is kind of centered here and there's nothing else around it, maybe your bin size needs to be different, right? Or maybe you need to scale your data so it's like properly spread out and stuff like that. So visualizing these different stages help you with the parameter choices. Am I making some sense? And then this last part is just drawing the graph. And then you can arrange the graph in any layout. I think it's like a circular layout. No, it's like force layout that he's using, Kamada Kawai, and you get this shape. And you can go even more fun and fancy with the D3, and you get this kind of stretchy, stringy thing that you can play with. Trefoil, not much to do, apart that it's a loop, and we got the loop. But when we apply this to fMRI data, it'll be fun, because then you can play the movie in time. You can see which nodes are highlighted, and I'll take you that step as well. Any questions about this part? Is it working for anyone? Do you guys try? Yeah.
We didn't accept the last step for some, I think that's neat.
Oh, okay.
Was anybody able to get the last step running?
The D3?
Yeah. I just had trouble on my browser.
Oh, no.
I was wondering if others had trouble there.
There's an error in the code
Where?
in the module.
Okay, not that I can debug it. I'm asking where as if I can debug it. Probably can't, but
I think it just was importing one of the submodules incorrectly.
okay.
Something weird.
Is it for everybody, the same bug? No way. Where did it happen?
There are 4. Sorry.
Yeah.
But it's still
Try to rerun it again.
Yes. So
Try the module, rerun it again.
do it again. Okay.
Yeah.
From the start.
Yeah, the last,
That's it? That fixes it?
yeah, that should,
Python.
yeah.
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 Record that. I mean,
The error seems to.
Yeah, rerun that model again, module that 4.4.
Oh, mine starts at 4.
sorry about that if that's still a
It's
problem.
not,
Is it working there?
get there?
The error is certainly on the see if it
Yeah.
makes it all the way.
Yeah, just rerun that, yeah.
There we go. Moment of truth. I think it's a browser problem.
You have Chrome?
No, I'm on Safari. Should I
Oh,
switch to Chrome?
yeah, Chrome, please.
Okay.
Yeah. D3 is not going to work in Safari, I think. Okay, so should we move to the next notebook or should we wait? What do you guys think? Yeah. Lauren, is it running for you too?
Yes.
So when you rerun it, does it work? But he's worked after rerunning it, apparently.
Something was in.
Okay, sorry about that. I promise you the MATLAB will be flawless because I know where to put the debug point. Okay, still in Python, this is the HAXB version. You may be familiar with HAXB dataset, right? Showing different faces, images, objects, and collecting ventral tegmental area in the brain, science paper. So we pull that data from NILearn and run mapper on it. That's the notebook number 2. Questions? It's notebook number 2 in the same GitHub. So I'll just do the basic running. Importing Kepler, dinosaur. Loading the data. We're going to load it directly from Axby. And I learn. Then visualizing the data. This is a time series from real fMRI experiments. So are you guys with me till this point? Is this one still stuck or working for you? Or?
I'm still downloading the data.
Oh, sorry. Maybe it's in my cache. That's why I downloaded so fast or something. I don't know. We can take it easy. Take your time, download it. Maybe I can come walk around. So we are on the HexP thing. Is anybody stuck? Able to get it? You keep installed and installed, right?
Just installed one, right?
Yeah. Oh, you have NI Sorry. NI learn. Do you have that library? NI learn.
I'll reinstall this again. Even this seems more.
Oren, is it working? iPad. I don't know if it'll work on iPad. Does it work on iPad? You can't run it. Can you? Okay, that's new.
She's having Python problems, but still stuff on.
Dinosaur, DYNEUSR. Is yours still stuck?
Yeah, still downloading.
Oh my God.
I just set it to 2 subjects.
Oh, yeah, one is enough. Yeah, okay.
It's like 30 seconds remaining.
Yours
Sort
working?
it out with like a 7-minute estimation. But
I'm just downloading the data.
yes, we have.
This is a network X.
Oh, like with the install just speeding up. So
Well,
I close.
okay. And you just did pip install dinosaur?
Yeah.
So it should have taken care of everything. It knows exactly version to get.
You run it in the environment.
I assumed that data.
You set up a new environment.
Perhaps like their records.
That could be it. If you already have installed a different version. So
Totally new environment and it's all about errors.
in network X,
Yeah, it ran.
it ran, right?
No, it didn't. No, sorry. It didn't run. So I created a new environment with just Jupyter.
Which part didn't run?
That's smart.
So do 3.6 as it says.
My
Yeah.
computer is so weird. I need to figure out like.
That's why I don't like Python. Sorry, guys. If nobody likes Python, let's go to MATLAB. Yeah.
Like.
Just like
Whoa.
activated the V3.8 instead of doing nothing anymore. The difference data open up there.
Yeah, yeah, that's what it is.
And so now it locked it.
Yes. Can you refresh it or does it go away?
Increment won't let me.
I was gonna just hope that
It's something the
so it's like.
so I can.
Let's just go to MATLAB, I think. I have a better version. And yeah. So I thought people here would be like Python fans or something.
Like 6 something for a lot of our like. Hopefully they didn't change something super stupid about this like function.
So I'm giving up on Python because many people are having, anybody was able to run this ish?
I am still downloading data.
Just download one subject, change the loop to like one subject. If you don't have to download 10. So if you're still struggling with Python, may I recommend going to the MATLAB version, which is also in Brain Dynamics Lab, but it's called, so you already have Dinosaur, you don't have to reinstall it. And new mapper is the new mapper, but in MATLAB. Let's use that one. We will still use Dinosaur for visualization because the D3 part is much cooler. Like the graphs you get from Python is still awesomer, I have to say that. But the MATLAB one runs without, hopefully will run without any bugs. So do you guys have MATLAB in your machine? And then just download the, and this you can just download like a folder and add any version. That's what good thing about MATLAB is. Any decent version should work. I didn't mean to sound so against Python. It should look something like this, the GitHub page, and you download the data, and I'll run it in MATLAB. So you download the entire folder as is, and we can run it in MATLAB by adding paths, blah, blah. This was tested in MATLAB 2020B. So hopefully you have more recent version, but 2020B and above should be fine. Let me know if you guys are ready and we can go to MATLAB. Are you guys ready to go in MATLAB?
Yeah.
Is anyone ready to go in MATLAB?
Sorry, I'm like,
So this is the website,
this on his GitHub, neuromapper.
new mapper, and then just download the entire zip or something, and open MATLAB, and then that's it. And you can add paths or go inside it, that folder. Anybody in MATLAB yet? So you can open within the new mapper folder, there's example folder, and there's a trefoil knot folder. And you can open the trefoil knot.m file. Yeah, there. And then if you just run it either cell by cell or however you want to run it, you should be able to hopefully it'll run without any bugs. But oh, there you go. Are you able to run the trefoil knot, the basic example? It pops up like this graph as well. Any bugs there? This one I can help with. Yes, no, maybe. Yeah, it worked. It didn't work? Yes, I am such a failure at demos. Oh, so you have in MATLAB this toolbox?
No, I guess.
Sorry about that.
That's okay.
Yeah, you need that.
Okay.
Yeah. Sure. Anybody else? Were you? Oh, you did it. At least one guy did it. Okay, good. Daniel, did it work for you? Do you have MATLAB?
Still.
Okay.
Oh, yeah, yeah, sure. It's probably, yeah, you've seen it.
Please fix it for us. It's not working. What's the problem?
So did you download this?
Did yours work?
Oh, yeah.
Okay, that's what Python does. It sucks you in. Anybody else tried MATLAB? Oh my God, Lauren, why did you get your laptop or something? Oh no. Lucina, where are you? We need a laptop. So you guys, anybody in MATLAB? Did you try MATLAB? You tried?
I did, yeah, I can get it.
It worked?
I didn't get it.
Oh, you didn't get that. Did it work?
It didn't work for me.
Where are you? Which file are you running?
I'm trying to run the faithful,
People not, yeah.
yeah.
New mapper, yeah. That doesn't
Yeah.
look like that file. Can you increase the brightness? I can.
oh, yeah. That's the create.
Yeah,
Yeah.
yeah. Yes, run this one.
And then just wanna
Oh, the one from the top.
change it.
Yep, change folder.
I know.
Oh, you need the toolbox. Do you have that one? Can you get that?
How do I get the?
So click on it.
On the phone?
So this is like the university MATLAB or something?
This one?
Yeah.
I'm on 2023.
So you have to like sign in and then install.
Oh, okay, I see.
I was running the tree portal.
Did yours work? Can you try?
I have it set.
These two.
It's working?
Is that some examples?
Show you.
Okay, so I'm assuming that the trefoil not work
Yeah.
for some people at least in MATLAB.
Then you have tree portal like that.
Similarly, there's another file, but this time in the Haxby decoding, the other example. And hopefully if we run this, it should work seamlessly, but if it doesn't, let me know. This is the fMRI data from Haxby. This is a graph. This is just a filter, and then this is colored by different objects that he had in the example. But that should run smoothly as well. And then the last part of that is it generates this Python file that you can visualize in D3. And you can do fun stuff. You can this is like take forever to just drag things. This is Haxby data, and you can also in background color, but you can actually play it like a movie. So this is where it'll start with TR1 and keep going. So this is the goal of today's tutorial. If you can reach here, I'll be very happy. So tell me where you are. Bonus point if you bring in your own data and crunch it. So anybody reach there almost?
No, it's like found that found.
Okay, can you go back to the gear?
Yeah,
I may have, and then new mapper and then example, and then XP decoding. Is the file there at least?
NPY file.
Maybe let me just add it. So if you go back to the first page,
Examples or
the new mapper, yeah, and then scroll down. I forgot to tell you, but he had a small script. Keep down, 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 down. Up, 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 up. Just about here. More up, maybe. So here, this is the script to get the data from NILearn.
Okay.
The NILearn didn't want us to put the data on GitHub
Mhm.
because it's their data.
Okay.
But if you just use,
Run this in Python.
yeah, it'll save that file, which I thought I did, but apparently it didn't go.
Okay. Yeah, it's good, but he said you have to run something in Python to download the data.
Let me try to put the data on GitHub. Sorry about that. Right. Okay, at the risk of getting some flag from NILearn, I uploaded the data on GitHub, so it's there. If you download it again, the new mapper should come with that data now. It has these TSV files now.
Mm.
Actually. So, sorry, download or sync or the new mapper again from GitHub. So you can have the data for HackC. And once you have that, then you can try playing with these parameters and see how it changes the graph. So where are we with MATLAB?
It was just the two TSVs, right?
2 or 3, I think. Timing and
Timing and onset.
labels and
Oh, timing and
one half, yeah. Labels and then this one. I don't know why then. So you
Oh.
just let them just in case. Yeah, 3-4 works here. Awesome. So now let's go to the example folder.
Yeah, so if you go to you can just re-download everything. That might be
See
easier.
the HackC? Yeah.
I just don't feel like
Double click on it because I just need some more files from the re-downloader.
Oh, yeah.
Are you guys somewhere?
Yes, now I'm able to get to the data.
Yay.
Oh, I see, data.
So did you do the trefoil knot already?
No.
Okay.
I just got the.
Okay, cool. Everything working?
Maybe I should just re-download it.
I can look in mine, right? I can just search, right? It should be somewhere in mine
Yeah, here because this,
to make sure.
look, I mean, here and I instructed.
You saw NIR, that's
Yeah,
good.
so this one, this node data function.
Let me check.
Okay.
Look, so it's getting it from NILearn, isn't it?
Yeah. Yeah.
So it's part of the NILearn. It's not.
No, I did this.
Oh, I
It
see.
was, it was.
It was like that.
Yeah, it was like.
From
that.
load data, I
Yeah.
got it. Okay. Yay. Let's go to number 2. And you can play with the one. You can try changing the resolution and stuff, see at what point it breaks. The load data is inside the dinosaur notebook.
So that
If you go to the dinosaur GitHub or within your load data is right there.
oh, okay.
Yeah.
So you just.
You got the data? Yes.
But no, it's saying it's why.
That didn't upload? Oh my God. Let me try again. I thought I did it, but I'm. Yeah, sorry, I just did add TSV. Let me add it again. Apologies. Okay, now try it.
Would be easier just to re-download everything.
Yes.
With the same file structure.
Very good. The example,
See examples.
actually, the NPY.
Okay, yeah.
Yeah, fine.
No worries, yeah.
Do you have the, so I just added one more file. Can you
Yeah,
download
I
it?
read on it. Should be the most updated one.
Can you go back to that? It has the NPY file. Do you see that?
Oh.
So go to the GitHub, like the vector.
Oh, yes. I just downloaded the.
Like just about like second ago. So go to examples or refresh it. A minute ago.
Oh, okay. Yeah. Worked.
Yes.
Downloaded.
Yeah.
Once it downloaded, it'll run.
Awesome.
Yeah.
And try the MATLAB thing if you have it. Otherwise,
I should. I'm sorry.
no, but it should work. It's still
Yeah.
Python. And email me if you.
Yeah, sure. Thank you.
No. What is the problem? You actually. No, I didn't see it.
Oh, yeah, that one. When it opens, if you just press run.
I wish I had Python expertise, I guess. Did you try the MATLAB thing? Is that working? You don't want to try MATLAB? That's okay.
That's right.
This is run it again. There you go. I think you ran the tree for, so go to the Haxley.
Oh, okay.
You got the NPY, right?
Yeah.
Doesn't show up? Yeah.
It might be the old download.
Oh, okay. Yeah. Yeah, I did it. Awesome. And you can also open that file. So go back to GitHub again.
Okay.
The web page. New app, yeah, scroll down. And just like, so because this, the next step is to save it as a graph so that it's in Python.
Okay.
So if you just run this in Python, in the same folder,
Yeah. Oh, in the same folder as the.
as you just did random MATLAB, except patchy hack, but then it gives you these pretty D3 pictures.
Okay.
And then save as an HTML. Hey man, you did it, man. I'm so happy.
Thank you.
At least one person got it. Yeah.
That's not.
Some people.
It's like fetching the state of set performing pre-processing.
Which is like very basic.
Exactly, but it takes time because it's processing, not just downloading.
I see. So maybe just do download one subject.
Yeah.
Don't do all things.
Okay.
That's a mistake, I think. We make.
Okay.
There are questions? Sorry.
Oh, yes, that's PNGC. And then, the wasn't .m, so whatever is .m is in MATLAB you had in the script. And then you can press play.
Take a second. Did it work for you?
So he said, yeah, this code.
This one, this one? Yes, yes. So just use Python. Like, how do you do Python? Is it in terminal or just type Python?
Yes.
And then copy paste, enter, or did it run?
Yeah.
That was too quick.
So I've got
I typically use like Jupyter Notebook or something. So easier.
Okay.
It doesn't have to be.
Anything, yeah. New one.
Oh.
But it has to be in the same folder where you the output of MATLAB.
Emotions, how do you remember things?
Wherever you download it. I think it should be in the example for the exam. Actually, and then do it here. It's actually already there. So if you go open this untitled, and then this one, not this one, the one below. This one just saves data. This one is something else. This one is. You have to have network. Yeah, network. Shouldn't be there. Which network X version do you have? Can you check? Okay. So go to terminal. And then Python, you're in Python, so import network X.
Is it?
No, just import space, network X. And then network X dot version, print network X dot version. In parentheses, print. And open bracket. We should do everything in me neither. Totally with you. Let's say. I think it's like underscore version. Something. So maybe try 2 underscore before that. Dot network X dot. Underscore.
Yeah.
Can you Google this? Yeah. Right. And then on the GitHub page it says which version of network X you should have. Let me Google.
Looking at this today. Am I not done? Okay. Imagine you are the dot, and then you become, you have to search for your own dot. Yeah, you're the proof.
Great.
I'm
With network X dot underscore underscore. Dot underscore version underscore.
Okay, I'm checking.
So if you go to GitHub, I think it needs 2.8.
Well, now there you can start a
The GitHub page for
astrology
initiating.
exhibition.
Because we are printing.
Exactly. Exactly.
Chronology. Tell you which one I have. Click on the diagram version.
I think you're the founding father of this new development. Elijah. Doctorate.
Let me see my version. We can install that one.
Yeah, so like, if you have a baby, you have to put it in the instant people play.
They will believe you.
Exactly.
So just post it.
Did you think about it? No, it's like, that's you just like.
Mine is 2.815. So if you go back to terminal. Okay, then get out of Python, just control D.
They would believe.
Say pip install, space install network X. Equal to 2.8.
So when it's literally just like standard, it's not even it's just like it's going.
Okay, now
So profound.
And then when he actually talks to us, like his actual study.
Yes.
That is the pain of stops. It's like he's working through.
Yeah,
So
he
really
has 3 kids, he had a job, and then he has to research them. I never do in my combined
True polymer.
Yeah.
like most of the times I feel imposter, but in his case, I just feel like.
let me see it with my code again. Did yours work? No, it's my bad. Does it work?
I don't I'm like, I don't even yeah,
She produced offspring.
Can you open a Jupyter notebook? Maybe then.
Yeah.
Yeah, I'm actually just learning this.
Yeah.
In the Python?
Yeah, I'm learning anything.
Okay.
Yeah.
Yeah. And then copy paste that code and as long as you're in the same folder.
Something that someone to do taking care of it.
Yeah.
Because they got married.
Yeah, you can just CD. Oh. Primary. Yeah. What's my last name at Stanford? So do you work in where do you work? Oh, cool. Nice.
Yeah.
Here.
Yeah.
You can put yours.
Academia, and I'm like, let's try through.
I got pumped. Dog. It's a lot of work. I'm all by myself. I'm good. So like, if I didn't publish 5 times this year, she's the reason.
You're a super mom. The mom is a model. She's publishing and then you're inspiring mom.
That's why I'm not publishing as much. Oh, then I didn't get nature.
It was very.
You give this picture.
Yeah, that's fine. That happens with me all the time. So I have 14 that I have a demo. I know. You know, this Python is very tricky.
He's not in the lab.
Yeah, the version
Start
system, you have to
Oh,
follow
yeah.
that, so then it works. Right.
No.
Yeah, you're going back today or?
They were in
Family
Atlanta.
here, spend some time with that.
Georgia.
Okay.
Now.
I see that his
Thank you very much.
Yeah, I see. But Judy.
Oh my God, you did it.
Yeah.
She did it. So we have one.
What's
person
going
who
on?
did it. Yes.
It's in MATLAb?
MATLAB.
Yes.
I got really going in
No
Python,
professors have.
but then the HTML kind of started freaking out. That's the only thing.
She was
that was
tired.
freaking out.
It's tired.
Otherwise, everything else.
Give us.
Yeah,
It's HTML embedding something that's.
yeah, exactly. That's the thing that, but putting Python in 3.6 was fixed it.
That fixed the network.
Like,
Like,
fixed it,
they're all
but
the same office.
3.6.
Something in the 3.6.
Exactly.
Yes.
It's not big anything.
Let's bring it up.
Appreciation warnings. And
So,
it's
All neuroscience awards. I might have some like an aluminum foil and
yeah,
grade mine.
So I ran
Awards
Met-
that was like from my first years.
Oh, that's the visualization.
Yeah, yeah.
I got this, but then
We're
trying to
working
do
Yeah,
the,
that's like I actually like the brand I did.
then,
So I
yeah, so I'm just
there
like
is just
running
something-
this.
But like, it's always
Yeah, it's getting
assuming that
there.
you're.
Like, I wasn't
just like
relatively. I'm sorry.
Oh, yeah. What version are you running?
Let's shoot again.
Switching it.
I'm sorry.
I got.
And it like blast the
No.
Set up an environment for.
right?
I haven't.
This is there, I was getting.
Doesn't do that.
Oh, but you're really not.
And all data that were in here. Which column?
Yeah, what?
We'll try to get through this. But you got,
Oh.
it's gonna be somewhere.
It's, you know, the hardest part is these HTMLs. If you're doing everything in Python, then
Should be fine.
it's much nicer. It's much nicer to you.
Yeah.
Um,
There was another.
Do you mind helping us here?
Oh, okay, I'll show you.
Yeah.
You go to my terminal,
Like what?
so like when I downloaded
So you're
this.
She solved it, so we should just take her.
The most sincere.
But I feel like I'm out here.
No, I wasn't, I think.
I felt
I'm not
more
sure.
correct.
Like something else.
Yeah, like a floating.
Like you don't fit in.
It's the same error.
Like that.
But
Yeah,
when I
that's
downloaded this.
Oh, the time was distinct. We'll have to regress it out like. Yeah, we're gonna have to.
3.6.
Sorry, I'm sorry.
Oh, okay, that's Python. But I think that might be it, because when I downloaded it,
He's not part of that
it was saying
Signal, yeah.
like some
He's not
version
part
of my.
of the
file is
what
incompatible.
background. I'm able to look at it, but I'm everywhere in the.
Year old. I'm just gonna have like literally just like a
Find it when
Made it, I'm hired now.
it's like an error.
I just feel, it was like, racist hit that people here.
Oh, no worries.
Wants to focus.
But the MATLAB thing gives
Yeah,
you,
yeah,
and that's
for the
basically
balance.
what you can define.
Like
This is all
black
fine.
ones.
Yeah,
That part is fine.
So-
but I like the one.
It's it's great for reviewers. Otherwise.
That's how we fool them. We can like picture. What is it? I don't know, but it's cool.
Yeah, it's suspicious.
Is it work for you?
No, I am.
Oh, that body behind
Something, something, it's like, it's keeping something in this cache.
Like,
Has
Yeah.
the parts part
I'll figure it out later.
Thank you, and let me know. Yeah, yeah.
She's
You-
posted all
Yeah,
the-
should I skip it?
There you go.
For now.
This will be Georgia.
Everything
Yeah.
seem intense.
Just go to
That's how you
MATLAB.
get the matrix.
There's a dead.
And then where we start. Oh, yeah.
Are they in the same building, just next to each other? The body is reminding you. To publish.
Yes, I got, I did install that. 2019 doesn't work. So what is it?
Right now, it's just reminding
2022.
school times when you have.
You guys get free MATLAB, right?
And if you're talking.
Yeah, no, I did install
Probably being
There's-
recorded.
2019. it was-
Oh, yeah.
So that's it, like, didn't matter.
I thought I could get away with it.
Yes.
Oh, that was very.
That's where the-
That
yeah,
means the class- We had one student who was like, didn't have, he used to be assigned.
And then you were saying trying to.
Like, it doesn't mean
That's great.
if I'm out of a class, you know.
Yeah, try to-
so students are-
Change correlation to you.
And I used to be there at four.
This one.
You're the deviation.
I was, and there were times I was the monitor.
Euclidean?
Did you actually write? Oh.
And then, oh, I see. And all the data's up here as well. Yeah, I uploaded it. You'll find it. Not necessarily there.
How dare you?
Were you at the principal's office again? Probably. I had gang wars in school. So,
I know you're.
If you guys are.
Thank you so much.
is you can, I was just saying, once you have this thing running, you can play with these different variables, like instead of correlation, try Euclidean or cosine. And this is it. So resolution is the number of bits. Remember, this is a new number, which means that it's binning on the graph. The binning is not as simple as just dividing the space into 10 by 10 spaces. So the first variable, the distance. So you take the point cloud, and the first thing you do is in this whichever space it's in. It's right now it's 2D, but whatever space it is.
Check once. Okay. So whichever space it is in, you run some kind of pairwise distances. Could be correlation, could be Euclidean, whatever you do. Now you get a distance by distance graph, a point by point graph, right? And then we do a reciprocal KNN, which is basically a K nearest neighbor graph. So the K in this MATLAB is how many neighbors to look for. So distance was first, so like which metric to calculate the distance between points. K is how many neighbors you should have. And after the resolution and gain is essentially, so you get the K nearest neighbor graph and you compute the geodesic distances. So as you guys know, we are computing the distance. So for example, if let's say this is your data, which has like an inner island and an outer island, it's like two rings, concentric. Then if you just do Euclidean distance, then these 2 points are this far away, which may not be true. The true distance would be you go from here all the way here, and then you reach there, right? That's what K nearest neighbor allows us to do, is compute geodesic distances. How many neighbors do I have to go through to reach there? And this is very important for nonlinear embedding. With the nonlinear embedding, remember that famous S shape, or I don't know if you do nonlinear embedding, like there's a famous pi spiral, like the distances are never Euclidean, they're always geodesic. And that's what we compute. And then we have a graph on that. We do this intrinsic binning where how many points should we pick? So you're given a graph to bin this graph into or compress the space. How many points should you start with? That's your binning parameter. So 400 means 400 points that I'm going to start with. So on the graph, I'll take 400 points. I mean, it has to depend upon how many points you have. So it might change, but about approximately 400 points. And at each point I'm going to blow up. And then that distance is this gain parameter. That parameter is gain. How much that should be blown up, like 40 neighbors to look for at maximum. And you connect these nodes in NewMapper if these blow up neighborhoods connect. And that's how you get the graph. Am I making some sense?
Are those defaults based off anything?
Yes, so in the NewMapper we present this. So for fMRI data, we did a bunch of parameter searching, and we found that based on the autocorrelation function and some other trivial properties of data, there are 3 characteristics we came up with. That's the algorithm look for in the data and comes up with those defaults. But you can play with them nonetheless.
And that was with the hours of data these were.
Yes, this is with like longer data sets. Yeah, but it's still fun. So let's try, instead of correlation, try Euclidean, just to see the graph. And this is how it pops up. And now let me just try the K, or actually this is way more fun to explore when you have trefoil knot. You know actually what the shape of the data is. So I'm gonna go from 40 to 20. So this is the trefoil knot with 20 resolution. If I go down to, let's say, just for fun, probably will break 2. Then what do you expect if your resolution is only 2? 1 dot. It's like 2 bins. So everything coalesces into just one thing. And if I go to, just for fun, 200. So this is something like, I think somebody was asking me about, event-related design or faster designs. This is something could be interesting for that. So if you have really high fine binning, but the gain is still pretty strong, it's going to kind of not artifactually, but it's going to, because the original number of points is very high, it's going to give you much nicer local structure that you might be interested in. So imagine an fMRI experiment where you have events, not blocks. Then having a higher resolution might help you as long as the graph can stay connected. So you can look at the local structure, how things are happening in like local parts of the graph. Is it making some sense? Versus capturing the global properties. The higher value relation give you more finer structure. Lower values gives you more global structure.
So the colors were the different stimuli in this one.
No, this is trefoil. So it's literally, this is the color, the input. The points are colored just so we know where red is, green is, blue is. They have no meaning in this case. So now if we go to Haxby and just for fun, change it to, I don't know, 40. Then as you would expect, you get a much, everything is connected to everything kind of graph. You can't really make sense of it. But if you go to, I don't know, 800. I don't know why it keeps popping up on the others, but then you get much more fine structure. By the way, the colors here are different object categories. I think blue is like resting states, nothing is happening, and these colors represent like, I forgot, faces versus scissors versus houses and stuff, so it's kind of separating them to some degree. But as somebody else said, nothing beats, what is that, the D3 visualization. It's worth it, even if it's not doing anything cool. So hopefully your Python code will work. Were you able to do that? Reach here?
No, you have to use Python to get to that part.
So after the MATLAB is done, then this last part.
Files. Python,
whatever that new map is. I think I like fully screwed up this time. But so here you have all the instructions. So it's like the trefoil not example. This is a Python code to get the Haxby data and then do the thing. And then you can have another Python code because it's a D3 programming. So it has to be done in Python apparently. Then you run this kind of code and it saves this HTML file, which has all the functionalities of playing it like a movie and stuff like that. It pops up. So once you're done with your MATLAB and it saves this mat file, .mat, go to the same folder and run this Python code, which should then spit out an HTML file that you can open up. So it's a very bad and patchy work. We need programmers to make it really cool, but we don't have it.
This is one subject's data in the Haxby paradigm.
Yeah, just the graph of that person.
And it's input as a
time series, region by time.
That's the NPY.
Yeah, NumPy file. Okay. Anything else? Should we call it a day?
Does anybody have any more questions?
I apologize profusely for this crappy demo. But feel free to reach out in email.
All right. Thanks, everyone. Stay good.
Yes, we do. And we do it on Sherlock, like our cluster.
That would actually.
So this new map. This is it.
Yeah. Oh, you got it?
Yeah.
But it's basically.
Because you did the 3.6.
You have everything in it.
So use Conda setup that it's on the GitHub.
Yeah.
Yeah.
And then do we ever ask you about this resolution? I'm sure we do.
Yeah, so I showed you, right?
Yeah, no,
Though there's something super weird if you run Python. You have to generate the file inside.
Okay.
Yeah, they usually use it.
My computer.
It's just that like one version. I think my issue, I don't know if everyone else was, because I have a higher version, it installed some NumPy, which was incompatible.
With the network.
It was saying like when I actually pip installed dinosaur. Yeah. There's
Oh, man.
most
Yeah.
functions are running except for
VT
one.
not why that's just each box those times.
I was like 1 year, I was like,
It's super, I know the old joke about all this stuff, it's like, it's free if you're not.
yeah.
Yeah.
It has to be in this.
Yeah, the people at MathWorks figured it out all the way back in.
I've experienced that.
But that's what they, that's, you know, professional.
No, exactly.
Right.
Honestly, like,
Oh,
I've always used it because I've always been
It-
So there's no need to put it in the gaps.
undergrad.
With ADHD and
But like, I know people complain that
That looks.
during like private. work,
6th year.
shit like that.
Even like.
Everyone good class?
Yeah.
And so.
I don't know.
But I always like to think that you have to, like, student license is very.
Yeah, I mean, like, it's free for you to.
Wait, what is this?
Like,
Right, right.
know,
Like
like,
1,452
What it
life
is,
forces,
like the one-
464
Yeah,
times.
it was like.
No, 464 R1.
no, I'm actually with Kat.
that's nice. I kept getting
Are you with
switched.
Tashia?
Like, right when you get, you got their trust, and then they'll be like, oh, you got it. I'm like,
I'm a big fan of
just
seeing this work.
go
Oh, she's
pay
great.
for the
Yeah.
phone.
I
And,
know.
I tried
He was
to.
like, something like-
Yeah,
That's
it's.
one of the games.
Yeah, I agree. I'll say, okay, you could be, okay.
Yeah, no, I just like, so I just.
Oh, and that was like.
Yeah, you could.
And the forward learning.
Yeah.
Yeah.
That-
I feel like Brandon.
Yeah, like, but you said, I don't care if you're giving me like, but I want
They're
to.
starting to leave the field because it's like
Like,
overwork.
And those are
Not the
the
pay is like
like
half the
like
thing.
super.
Yeah.